Okay, welcome everybody. I apologize for the slight delay. We had to work out some technical issues, but we are all here for your entertainment, so it's going to go fine. Okay, so I want to start by welcoming everybody to the 8th PEDS Protein Engineering and Design webinar. My name is Roberto Chica. I am the editor-in-chief of the journal PEDS, which is short for Protein Engineering, Design, and Selection. And before we get started with the webinar, I just want to make a short announcement. So I'll share my slides. Okay, here we go. So as I mentioned, I'm the editor-in-chief of PEDS. This is a journal that's published by the Oxford University Press. It's a not-for-profit journal that was founded in 1986 by and for protein engineers. And over the decades, it's published the work of almost anybody that you can think of in the field. So thanks to this, we can draw from a large database of expert reviewers for your work. And our review process has been streamlined since last year, so it's now faster than it's ever been. We have an average time to first decision of just over 4 weeks now. And we have no publication charges except for the optional open access fee. So if you have interesting protein engineering and design research that you'd like to publish with us, please don't hesitate to submit your manuscripts. And if you want to know what we've been up to, you can follow us on Twitter using the handle indicated here. Okay, so now that I've told you about this, I just want to say a few things about the webinar. So I'd like to start by first thanking Oxford University Press for helping me put together this webinar. In particular, Sarah McKenna, who's been handling all the logistics behind the scenes. She's been, without her, I wouldn't have been able to do this. So thank you, Sarah, for all your hard work. It's been a pleasure working with you to organize these webinars. And I will also mention that this is the final webinar of the first season, if you want, of webinars. We'll be stopping through the summer, given that there's many online conferences that are starting soon. Okay, so for those of you who have attended these webinars, you know how it goes, but typically we have a short 20-minute presentation by, in this case, a member of the Editorial Advisory Board of the journal, Dr. Bruno Correia from the École Polytechnique Fédérale de Lausanne. And after that, we'll have a short question period. When you want to ask questions, you can type them using the Ask a Question button that's at the bottom part of your screen. So please type them there. And Dr. Correia will answer your questions after his presentation. After that, we'll move forward with our second presentation, our keynote presentation from Dr. Liz Meiering from the University of Waterloo. That presentation will last for 40 minutes, followed by another question period. And if everything goes well, we'll be done in about 90 minutes. Okay, so without further ado, I'd like to introduce our first speaker, Dr. Bruno Correia. It's a pleasure to have you with us today. So Dr. Correia is from Portugal. He earned his bachelor's degree at the Universidade de Coimbra in Portugal. And from there, he moved to the United States, where he worked at the University of Washington for his PhD, working with Bill Schief and collaborating with David Baker. And when he was a PhD student, he worked on immunogen design and vaccine engineering. After that, he moved to California, joined the Scripps Research Institute as a postdoctoral fellow, working with Ben Cravat on the development of novel chemoproteomics methods for the identification of protein small molecule interaction sites in complex proteomes. And since 2015, he's a professor at EPFL in Switzerland, and he's developed a vibrant research program, still developing methods for protein design and performing immunoengineering, and more recently, starting to use machine learning and for the prediction of protein-protein interaction specificity. And in 2021, he's the winner of the Protein Society Young Investigator Award, a highly prestigious distinction for early career researchers. So it's a pleasure to have you with us, and I look forward to hearing your presentation.
All right, great. Thanks a lot, Roberto, for having me. Thanks a lot also for doing this service to the community, you and Sarah setting up all these webinars that have been kept people together. So without further ado, I will tell you about some of the research that we've been doing here in Lausanne. And I will tell you about how we're using computational methods to expand the functional scope of computationally designed proteins. So today, essentially, I will actually be very narrow in scope, and I will focus mostly on the applications that we've been working on for immunogen design. So please click, Sarah. Yeah, so, and again, Sarah. So yeah, but just to tell you another, how our lab actually tackles the problems in terms of protein engineering. So we have a component on protein modeling and methods development, as Roberto was mentioning, where we work in terms of pushing the methodologies to design proteins with novel functions, particularly those that still remain very, very challenging. And then on the other hand, most of our projects also always have a very strong component in terms of biochemistry and biophysics, and using high throughput screening techniques, and in some of these projects actually in vivo testing. So please, let's go forward. And so you can read some of our papers, but today, as I mentioned, I will only tell you about the work that we've been doing in designing immunogens for vaccine development. Please go ahead. Okay, so again, to remind you, to remind you, the slides are having a little issue in terms of formatting because we had some technical problems, but I think you can still see them. So in terms of reminding you, what is the typical problem of a protein engineer, is really this problem of space, right? So we do have these issues of an almost a virtually infinite number of combinations, where when you start very quickly going to proteins of like around 100 amino acids, you have a potential number of combinations of polymers or proteins that you can make of, more than the number of atoms in the universe. And interestingly enough, as the genomics efforts have been kicking in, we actually see that space has only really explored a very small part of, or nature has only explored a very small part of that space. Please click, Sarah. But if we want to simplify and think about how the protein design exercise actually works, most of the computational protein design community, actually what we do is we start with a template of a structure, in this case, the backbone of a protein that you see here shown in yellow. And then computational algorithms are used in order to try to explore some of this space in silico, and then you'll have the generation of different sequences, and of course then these sequences, please click, then you can either ask a number of questions once you start engaging in these design efforts. If you have more the fundamental design principles in terms of trying to understand how proteins actually work and what are their determinants for folding, or a more biotechnological type of interest, where we start getting engaged in design protein-based drugs, enzymes and biomaterials, for instance, amongst others. Click, please. Okay, and so typically the computational protein design algorithms work, broadly speaking, in the way that I'm showing you here in this slide. So typically you have a force field, a protein architecture definition, and some type of user-defined constraints, either on sequence or on structure. You then start sampling different backbone configurations and sidechain configurations, and using the force field, then you rely on a scoring function to search for the better energy, and the lower energy solutions, or sequences in this case, will be then further characterized. Next slide. So the challenge, of course, of designing function in computational design is one that is very important. And if we click again, a lot of efforts have been doing in terms of de novo protein design, but what has been relatively challenging, and now I think we start doing noticeable breakthroughs, is in fact in terms of installing function into this de novo design proteins. So one more click. So of course, we have been very successful in designing structurally accurate proteins, but oftentimes they do lack this functional component. Next slide. So thinking about how to design function into proteins, here in this diagram, what I show is basically two approaches for this same problem. So while on one side, on the left side, you have basically this idea of a top-down protein design approach where you can start from the natural repertoire, you then use the functional motifs that you see in the middle, and you end up with functionally repurposed proteins. On the right side, you have a more bottom-up type of approach, which we refer to bottom-up protein design, where you actually start from building proteins just with the structural domains like helices and strands, and then you assemble topologies, which ultimately can embed, again, these functional motifs, and ultimately what you end up with, as you see in the top of the diagram, is the functional de novo, what we call functional de novo proteins. Click, please. So basically what the design objective here is going to be in this, in the next slides that I'm going to show, is about functionalized de novo proteins with binding sites. And we have used these types of proteins for a number of different applications. So some of them for biosensors, to create biosensors to profile antibody responses, to create bifunctional proteins that you can use to control signaling in cells, or for vaccine immunogens. And vaccine immunogens is where I'm going to spend the majority of the time. Next slide. So, but this approach of bottom-up de novo protein design, here I will introduce you this algorithm that we call the Topo Builder. And the Topo Builder is nothing more than an approach to sample protein architectures at different levels of resolution, where you can start from, you see from your left, right, by defining the architecture of a protein simply by its 2D topology, where I'm showing how you can define, for instance, a 4-helix or a 3-helix bundle in this case with a loop. And then on the next step, you have a geometric analysis stage where you start connecting the secondary structure elements, and then you translate these 2D diagrams for the 3D space. And in the next slide, hopefully, Sarah, if you can click and play the movie, that would be great. Exactly. I'll give you a better idea of how this actually, well, it might not actually work, but ultimately what's going to happen is that we will fold the proteins around the functional sites of interest. And ultimately, if I can try to make this work without having a movie, is what you see on the bottom of this slide is you have the purple motif, which in this case is the functional portion of the protein. And you will fold the proteins around this functional motif, and you will end up with proteins that have slightly different topologies, but still present that functional motif. So next slide. And we have done this for a number of different topologies. And I think here in this slide, you can also see what types of proteins can be designed. So we have tried this for alpha beta topologies, which you see on the left. We also have full alpha topologies with complex binding sites. And then further on, we actually push this to embed into this de novo proteins two distinct binding sites. In this case, these binding sites are structural motifs that are recognized by monoclonal antibodies, and you'll see what the motivation is to do this on the next slides. Please, next slide. But the way that we do this is not purely a computational approach, but rather one where we use computation together with high throughput screening and library generation. And ultimately what we do is we take the best designs out of this in silico protocol. We assemble libraries based on these designs. We use yeast display, and we expose these libraries of designs at the surface of yeast, both to binding selection pressures as well as to folding selection pressures, which in this case is related to protease resistance. And then using a next generation sequencing approach, we figure out which proteins or which sequences have the best characteristics. Next slide. And so we have done a number of different designs, and once we have this design in hand, then we can actually express these proteins and characterize them biochemically. So the main messages of these designs is that ultimately many of them seem to be well-folded and stable proteins. And more importantly, what you can see in the last row of this figure, in this biocore data that has the traces in red, is that these proteins also seem to be recognized very clearly by the monoclonal antibodies. Next slide. Then, as a last stage for judging the accuracy of the design protocol, I show you here 3 different crystal structures that have been determined and that have been compared to the computational models. And what you see is that ultimately the differences or the similarities between the computational models and the actual experimental structures are all within around 2 angstroms, which means that these proteins are extremely similar to one another in terms of the model and the experimental structures. Next slide. Now, what I want to tell you is about what we've been doing with these proteins, particularly in the area of designing immunogens. And our approach is based on what people refer to as the reverse vaccinology approach, and that is one of the most used approaches for structure-based immunogen design. And what this approach refers to is that we typically learn from immune responses from human patients, in this case, where the antibodies are isolated, and crystal structures of those monoclonal antibodies in complex with the proteins of the antigens are solved. And once we have that structural information in hand, we can then use computational design to engineer better immunogens. Next slide. And so, and maybe you are wondering why this is actually relevant to do, although we are in the middle of a pandemic. But the idea here is that there's this one very important challenge in vaccine design, which is how to focus immune responses. So let's say that you have, for instance, in this case, I'm giving you the example of a viral protein, which is hemagglutinin at the surface of the influenza virus. And one of the domains is very variable, which is shown in green, and the other domain is very conserved. So if you would have a perfect immune response, all of that immune response would actually go to the conserved domain, since then the virus would have less room to come up with escape variants. Next, please. And so the idea here would be that we actually are able to design antigens that can bring the attention of the immune system towards particular sites which are more conserved than other sites in the viral pathogen. Next slide. And so, and for this, there's a number of different other strategies that have been used. But today, of course, I will tell you about our efforts in using de novo protein design to achieve this. Next slide. And so the model system that we have been working here in the lab is the respiratory syncytial virus, a virus for which there's still no vaccine available. But what there is, though, is something that for us is very important, is a very deep structural knowledge of the viral protein and also of the types of human antibody responses that the human immune system actually mounts. Next slide. And so actually, when we compare the types of immunogens or the de novo immunogens that we've designed with the epitopes that are actually shown in the viral proteins, we can see that they're actually very, the structural mimicry is very close. And that's essentially what you see here for these 2 epitopes that we have designed de novo proteins for. Next slide, please. And so to give you an overall idea of where this project started and where actually it took us, we started by extracting epitopes that were recognized by neutralizing antibodies and using the topo builder in order to create novel stable immunogens. Once we had these novel stable immunogens, we could then test them in 2 animal models and analyze the types of antibody specificities that we were obtaining from these immune responses. And this is what I'll tell you in the next few slides. So we started by doing tests in mice. And so here in fact, I'm showing you 3 different antigen groups. One of them, which is a Trivax2, just immunized with the de novo designed antigens. A second group, which is labeled the RSVF, which is just a positive control group used that was immunized with a viral protein. And a third group where we prime the immune system of these mice with the viral protein. So, and then we boost with the computationally designed immunogens. And here we're mimicking an immune system that is non-naive and that has been primed, for instance, with a virus. Let's say that you have an initial infection. And so what you see here on this plot on the right, where you have RSV neutralization, is that, in fact, just on the Trivax2 group, we were able to already obtain 6 animals out of 10 which showed neutralization activity on just being immunized with the computationally designed antigens. You see, another thing that is also very important to notice is that this neutralization activity is 2 orders of magnitude lower than that you obtain with the viral protein. And of course, I'm happy to discuss this in the question session. Next slide. What we can see, though, is that when we immunize these mice with Trivax2, and so for this plot, which is a little bit loaded with data, is that most of the response that we are obtaining is, in fact, focused on 3 different sites of the viral protein. And so these are the sites that you see shown in purple, yellow, and red. And while once you have an immunization done with a respiratory syncytial virus protein, the F protein, in fact, you get responses that go against many, many different sites of the protein. And so this was one of the first evidences that we were actually able to create an overlapping response, that it was very focused towards the neutralizing epitopes that we were targeting. Next slide. Then we set out to ask the question of what would these immunogens could do in an animal model that would be close to humans. And in this case, we actually immunized non-human primates. Again, you have here the immunization schedule. From this first piece of data, what we could see is that we could, in fact, elicit antibodies that could cross-react with a viral protein. So that's one of the first requirements to understand the functionality of these antibodies. Next slide. And then, of course, we also checked if the response was focused towards the antibodies towards the epitopes of interest. And what you see in this plot is also that all the animals developed the responses that were mostly dominated by what we call the epitope of site 2, that had a large fraction of reactivity in this immune response. And next slide. But more importantly, 5 out of 6 animals, in this case, were able to develop a neutralization response. So now we obtained a much better neutralizing response that we had obtained in mice. And this, of course, it was also not very surprising to us because we had seen this before, but nevertheless still shows us that mice are not macaques, and they're also not human. And there's certainly differences in between what different immunogens can do in different immune systems. Next slide. So lastly, I will just go very quickly over this. We also studied at the molecular level which type of antibodies were being elicited in this non-human primates. And so we isolated 2 different antibodies, one of them called RSV C57, the other one RSV C19. Next slide. And what we could show is that one of these antibodies was, in fact, directed to one of the most important epitopes in this protein. In this case, was a site 0 antibody, and these antibodies were also able to neutralize the virus as monoclonal antibodies. Next slide. And more important was that when we actually compared, or we asked, well, is this antibody actually similar to one of those that is seen in humans upon natural infection? Next slide. In fact, we could see that this was this antibody in its sequences had features of one particular lineage that has been seen in humans, and that has been documented to be present upon natural infection. And basically from here, what we could conclude was that through this vaccination with this de novo design antigens, we were indeed able to elicit neutralization antibodies that were also elicited by natural infection in humans. Next slide. So with this, I'm very close to the end, and I would just like to conclude the talk until here. And so bringing you back to the beginning, what I've shown you here is that we can use the Topo Builder to generate functional proteins, particularly in different types of topologies with different types of complex epitopes or functional sites, as you will. We've used this for both to create proteins which or to create functionalities in proteins which generally didn't exist before we made these proteins, and we focused on the demonstration of the use of this designed proteins as vaccine antigens, and here we show that there was the induction of epitope-focused responses, and also of the elicitation of neutralizing and human-like antibodies. Next slide. Now, of course, this all sounds, I think, good, or at least it sounded good to me in many different times. Sometimes it also doesn't sound that good, but often what I think that it's still kind of unfulfilling about a lot of the things that we're doing is that part of it are still very manual in a way. So we would like to have more automated pipelines to help us design these proteins, and we would like to have much more clearer metrics for the quality of these designs in order to help us, for instance, using perhaps a Build-A-Grado language to understand what the designable scaffolds are, in the sense that if we could recognize ahead of time what type of backbones and what type of configurations of the scaffolds would give us the best sequences, then perhaps we could narrow down much more the choices of the sequences that we would test in vitro. And so, and I think here there's still a lot of work to be done in terms of more methods developments to make this process more automatized. And of course, we're now excited about trying to apply this to design antigens against other pathogens. So, and I think now it comes the last slide. Next slide, please. Which is also the most important slide. And so I really would like to thank all the people that have worked with me here in Lausanne. This has been really a great time of my life until now, and I've been very fortunate in having had a really talented and motivated a number of people that had shared a number of good moments and also bad moments, together in this ride that we had in trying to make these things work. And also, of course, a word for the funding that we have obtained throughout the years. And with this, thank you so much for your attention. Very sorry for all these technical problems that we were having, but I hope that the message still came through, and I'm happy to take any questions that you might have.
Great. Very nice talk, Bruno. That was nicely done. And I like the teamwork between you and Sarah there.
Critical.
Okay, so I think there's already a question for you in the Ask a Question button. So I'll ask you to read it and answer. And please don't hesitate to ask more questions if you have.
So here we go. So the question is the following. Will the lowest energy configurations always be the most optimal form of a protein for a protein precursor that is used for further design and optimization? So that's a key point. And in all honesty, the answer is partly no, partly we don't know. But what I want to say is we have looked into, actually very deeply into all the features of the models of sequences that seem to be stable and gave us good binding interaction or good binding affinity. And we could not encounter this indicator of energy, that all of those were the most stable proteins according to at least the scoring function that we were using within Rosetta. So what does this mean? Ultimately, it does mean that I think here the optimization of these designs is also twofold, right? Because we are not only optimizing for stability, but we are also optimizing for binding. And I don't think that necessarily the proteins that are going to bind the best are the ones that are going to be the most stable. So I don't think it's also expected that the more stable designs are the ones that are going to be the best binders. I hope that I answered your question. Then there's a second point. Can you comment on prospects for design all beta proteins? Yes, so I can definitely comment on those prospects. And we, of course, have been trying to do our efforts as well. I think the prospects, sadly, are not very, very encouraging yet. But I also don't want to be negative. Okay, so I think that there are a few indicators that it can be done. But right now, I do think that, for instance, in all beta proteins, there's 2 points which for me are critical, which I don't think we have fully under control. One of them being the aggregation propensities, which are typically present and latent on the edge strands. Okay, about that, you can perhaps go and perhaps do some negative design, or you work those edge strands very carefully to avoid that they have this tendency to aggregate. So that's one way, and I think some work has been done in that direction. But I think there's one that actually is a lot more interesting to me, which I have no solution, but I would love if people would have like ideas, which is about this way of how do you actually design proteins to fold with long-range contact interactions. And let me see if I can understand this, if I can explain this very well. I think generally we're really good at designing proteins that have short-range contact interactions, so proteins that are very local in contacts, right? So you have helix 1 that touches helix 2, and helix 2 touches helix 3, and this is all good and fine, right? Because then I think when you start designing towards a low energy minima, it's kind of like a puzzle. Everything falls into place. Now, when you have proteins which have long-range contacts, I think that there's a number of aspects that our computational methods don't account for, right? Because our computational methods, the only thing that they see is a folded configuration of a protein, and they design to stabilize that, but there's really no notion of the folding path. And I think this is, this to me still remains an unsolved problem in computational design. And I really would love to have more discussions with people to understand how we could actually encode or include in our computational design methods this ability of having specific long-range contact interactions. I think this is a fascinating problem. And I think this is part of the problem for the all beta proteins. Okay, long answer for a short question. So we have another question. Can you comment on conformational changes upon binding? Yes, I can. Not necessarily that I can comment a whole lot about our own proteins, because I think our proteins, for instance, we have designed them to have pre-stabilized productive conformations of binding. So, the whole design process in us is biased to stabilize one specific configuration, which is the bound state. So in our sense, we basically try to avoid that problem because we also don't have a good way to solve it. But in this way, we know that by pre-stabilizing the configurations of these epitopes or binding sites, if you will, we do gain in terms of binding affinity, because then you no longer have a conformational entropy, which is not very productive for your binding oftentimes. Now, one thing that I cannot tell you is if, in fact, for instance, in our designs, we see induced fit from the binding interaction or not, because we only have one unbound structure, which was solved actually by NMR. And certainly the the binding site doesn't look like exactly how it looks like in the bound interaction. That said, this binding interaction that we had was a binding interaction of like about 125 picomolar. So like something pretty tight, okay? So, it's not like there was a lot of room to have many different configurations, because it probably also works very closely like a lock and key. So right now, at least from our standpoint, as far as I can tell, mostly we are stabilizing bound states, and I don't think that we're having a lot of changes upon binding, but they could happen.
And Bruno, maybe I can ask a question. So I can see the advantage of Topo Builder compared to the Lux method developed by Tanya Kortem's lab, because you're not restricted to helices and loops. But I wonder if you can comment what the advantage of Topo Builder is compared to the mini protein design methods developed by David Baker.
Oh yeah, sure. Yeah. So I think the mini protein designs were in a way a little more naive, or okay. I mean, maybe they were probably, free subjects in life. They had no constraints, right? As long as there was a core that this secondary structure elements could form, this would be okay for these mini proteins. I think in our case, we're restricting the possibilities of these topologies by a lot, by imposing that they have to present a particular binding motif. And oftentimes, whenever we're talking about this binding motif, it's not a secondary structure regular binding motif. Some of the ones that I showed you, they're actually binding motifs that are presented by two different discontinuous segments, right? So in a way, you can see the mini proteins as like, free floaters. They're free. They can do whatever they want as long as they're stable. In our case, these proteins would not be able to do whatever they want, because they still have this very strong constraint of presenting a structural motif for binding.
Right. Okay. Thanks. All right. Great. So I guess we'll stop here and move on to our next presentation. Please join me in thanking Bruno for his excellent presentation. Okay. So our next speaker is Dr. Liz Meiering from the University of Waterloo. Dr. Meiering earned her bachelor's degree in chemistry from the University of Waterloo, and then she moved to England for her PhD at the University of Cambridge with Alan Furst, my esteemed predecessor as editor of PEDS, and obviously a giant in the field of protein engineering. Then from there, Dr. Meiering moved to Harvard Medical School to work with Gerhard Wagner, studying protein biophysics. And since 1996, she's been a professor at the University of Waterloo, where her research program is focused on protein biophysics, protein folding, and protein engineering and design. And she was previously a university research chair at Waterloo, and in 2019, a visiting professor at the Weizmann Institute of Science. So it's a pleasure for me to welcome my colleague, Dr. Meiering, and I look forward to your presentation.
Thank you so much, Roberto. Sitting quietly. Got to warm up the voice. So I'd like to also thank you for this invitation and for creating this forum for us to talk about protein engineering. I think there's so much exciting work going on, and I think I look forward to this network continuing to grow, because I'm sure proteins are going to have, engineered proteins, designed proteins are going to have more and more of an influence on our lives. So you can see here why I asked Bruno the question about beta proteins. One of the things that we've been focusing a lot on is beta proteins. And of course, they're not always very well behaved compared to more helical proteins, particularly when you try to design them. So our research has gone increasingly towards understanding their solubility and self-association and what the mechanisms of aggregation are. So I'm going to talk about 3 different proteins today. First, I'll talk about this designed protein. It's a 3-fold symmetric protein that we call 3-foil. Then I'll talk about ADNectins, which are also known as monobodies, which are engineered binding proteins. And then superoxide dismutase, which is associated with causing ALS. Let me just make sure I've got, there we go. Okay, so, all right. So I've kind of given you an overview of the 3 proteins. The problems of solubility and self-association are widely impacting any applications for proteins, whether you're doing fundamental research, whether you're doing applications, for example, purified proteins that you might want to make as a drug or for biotechnology. You can have these things form native-like aggregates or amyloid-like aggregates, and typically you want to avoid this kind of aggregation. Another new field that we've become also interested in is inclusion body formation. I'm going to talk about that. So with inclusion body formation, many of you may know that when you try to make your recombinant protein, it can end up in a big body that you can see by microscopy. And very little is known about the structure of this, so I'm going to talk a bit about that. And inclusion bodies are also increasingly becoming of interest as a material that you could engineer for controlled release of proteins. So if you can understand how they form and how they, what stabilizes them, how they redissolve, that's, I think, going to be another interesting area to pursue in the future. And then here on the right is a messy view of what solubility, self-association, and aggregation looks like from a review by Kitey and Dobson. And proteins can just misfold in a whole bunch of ways. They can make amyloid, which you may be familiar with. They can make amorphous aggregates and native-like aggregates and interconvert. So it's a hard problem, and I think there's a lot still to be done. Okay, so in terms of trefoil, this was a protein we made a long time ago now, and we were using at that time consensus sequence, internal consensus within the 3 repeats of the protein within the polypeptide chain, also consensus with close homologs so we could retain function. We used homology modeling and Rosetta, and we made this protein that had 3 identical repeats within the chain. And we were delighted that although we changed 30% of the template to make this thing, we got it on the first attempt. And we got a lovely crystal structure that was at high resolution, 1.2 angstroms. The protein was functional, and we thought, wow, design is possible. So what I'm going to tell you about is how we followed up on this, because I think it's really important to follow up, and the devil is in the details, and we were hearing about that in the first talk as well. So what we found when we started to characterize the protein more is it has extreme kinetic stability. So this is the rate of unfolding and folding, which are correlated with each other. Trefoil's over here. It folds and unfolds much slower than other proteins. And for context here, its unfolding half-life is about 8 years. Its folding half-life is about an hour. And normal proteins generally fold and unfold much faster. So we made something that we were surprised to find had these properties. And so we characterized it further, and we found that kinetic stability correlates with other kinds of stabilities, such as stability against proteolytic degradation or against unfolding by detergents. So here you can see a gel of where we have trefoil incubated without protease. So the protein is 1 mg per mil, the protease is 0.1 mg per mil. We leave it for a week, and trefoil only loses its his tag. By contrast, these other proteins are almost all degraded. And the exception here is SOD, which I'll talk about later, which in its mature form is extremely stable. So this was interesting, and we wondered how general might this be? And so here we're looking at all the data we could collect from the literature, as well as screening for kinetic stability that we did in collaboration with Freddie Colin's group. And what we found is that the absolute contact order, which is the average separation between residues that are in contact in the folded protein, is correlated with how resistant the protein is. So here you can see when we classify proteins into not resistant against detergent denaturation or resistant, we have a higher contact order, meaning more long-range interactions and a larger distance between interacting residues. It's not an absolute trend, but it does tend to occur. So I think this comes back to what Bruno was saying also about getting cooperativity in long-range interactions is an important thing to think about. So what we did is we looked at this in more detail in collaboration with Shachiko Salvi's group, who does coarse-grained modeling. And I'm just going to highlight here a comparison between trefoil and symfoil, a threefold symmetric protein that has the same topology, no detectable sequence similarity, and a different kinetic stability. It's less stable. And here are the structures superimposed. They differ in these red residues, which are in functional loops for trefoil. And we could look in more detail because we have quite a lot of scatter. If you just use average sequence separation, that's not a brilliant measure of complexity of the structure. So with coarse grain modeling, you can capture more of that. And we found that the barrier for trefoil was indeed much higher than for other proteins of the same topology. And if you remove these interactions that these loop residues make, which tend to be long range, the barrier comes down significantly. So having these many long-range contacts is important for creating this high barrier and perhaps this cooperativity that also gives us the other resistances against protease and detergent. So we are actually following up on this to try to engineer kinetic stability as well, but that's for the future. Okay, so then this is kinetic stability that I've been talking about. We also were interested in further engineering function, and we wanted to make a more stable protein to start with. So we thought, let's try to increase the thermodynamic stability of trefoil. And so we thought this should be easy. There are many, many predictors. We will combine all these predictors. We made a meta predictor, which appeared that it would have good accuracy in making a stabilized protein. So what did we get? In fact, so we picked these 10 mutations that are all around the surface of the protein and that were predicted to be the most stabilizing and that didn't mess up function. And what we found is, if you look at this, we got something that was stabilized. The green ones are the stabilized ones, and the light gray is predicted, gray is experimental. So we got some stabilized variants, but not very stabilized. And you can see the nasty orange ones here. The ones that were predicted to be the most stabilized were in fact destabilized. We can talk about that. We think that hydrogen bonding was one of the problems here. Nevertheless, we could combine these stabilizing mutations into these multi-mutants, and we got considerably stabilized proteins. And if we compare the accuracy or the likelihood of getting a stabilizing mutation, if you do this randomly, the probability is low. And if you look at mutations in protherm, we predicted, should the mutations that are in that database be stabilizing or not? You can predict it reasonably well, and we did better with our meta predictor. So you can stabilize proteins, but it's not a sure thing. I think we can get things roughly right. That's kind of a theme. But precision we don't yet have, and we need to improve this still. Okay, so we made these stabilized mutants, and this brings me to the solubility aspect. What we found is that compared to the wild type, the single mutants were mostly good. The slightly stabilized ones maybe even were a little more soluble. But the mutants, the point mutants, all started to tend to be less stable, Q78I in particular, and our multi-mutants are terrible. So this was a hassle in making folded protein because it actually expresses in inclusion bodies. And when we looked at it after the fact, we went, well, yeah, no wonder they're bad. They're mostly making the protein more hydrophobic. You can see this. And so we have removing a charge, adding aliphatic hydrophobic side chains. And you can predict this to an extent with a variety of predictors. It's not very precise, but you can rationalize it. Okay, so hydrophobic mutations are there. They're messing with the stability, with the solubility, but they are actually making it more stable. So we looked at this in a little more detail. And actually, first, let's talk a bit about stability before I come back to solubility. So we looked here at mutations that were designed to be, or predicted to be, more stable. And we collected the data from the literature and compared with what happened in experiment. And what you can see is that on average, mutations that are predicted to stabilize are about neutral. You don't generally tend to get increased stability, but a fraction of them are stabilized. If you look at the different methods that were used to design increased stability, you can see they all have a problem. Blue is generally higher than gray. And if you look in a little more detail, what you can see is that the agreement tends to be better for statistical potentials with machine learning. So those seem to do a little better, and consensus design works. So I think this was actually an aspect of why our original design worked as well. We don't know why consensus design works, but it does seem to work. Okay, so you can stabilize, perhaps better with some tools than others. So now let's come back to solubility. When we looked at all the mutations that had been designed and calculated their change in solvation, or in other words, their solubility, and grouped them into the stabilizing mutations that are shown in the blue and orange that are the mutations that destabilized. What you can see is that the orange bars are typically lower than the blue bars. In other words, the stabilizing mutations are generally less soluble. So, less soluble is a bit of a surprise. One thing to note again is that exception here was consensus design again. So here, the ones that were more stable were also more soluble. So what's going on? Why are these mutations behaving this way? So I'm going to re-examine what goes on with accuracy to give you some context. So we were using Protherm data. So this is a curated database of stability of experimentally characterized mutations. And what you can see here is that there's only a small proportion of mutations that are stabilizing. And many of these algorithms that predict stability are tested against Protherm data. If you break down and look at what is the accuracy of prediction with these different stabilities, what you can see is we get pretty high accuracy for the destabilized variants, which, by the way, are often truncations and in a core. But in terms of stabilizing mutations, the accuracy is much less. So we have a challenge in predicting stabilizing mutations. Okay, so given that stabilizing mutations are hard to design, why do all these designs work? And so this is an attempt to partially explain that. So if you look at the distribution of stabilities for design mutations, we said it was centered design mutations that are designed to stabilize. It's centered around 0. If you make multiple mutants, that distribution spreads out. And so that means now you have a possibility to make considerably stabilized variants when you make multiple mutants. And so if you want success in your designs, maybe these point mutations that we were making are, not so likely to work out and achieve your goal. But if you have more mutations, because you tend to get each one roughly right, once you do many of those, you're bound to get some good ones in there and you have a higher chance of success. And you can get 3 kcals per mole stabilization with a relatively small number. And with more mutations, you can get very strong stabilization. Now, so that might explain a bit why we're having design successes when you make many mutants. Another thing to note here is if our prediction accuracy increased even a little bit from 0 here in the solid line to 0.1 kcal per mole stabilization, you would gain a lot in terms of success of making stable proteins. So I think our prospects for the future are good here to continue to improve our design success. Okay, so that was trefoil. Now I'd like to switch to discussion of ADNectins. And so ADNectins are a scaffold, a binding scaffold that resemble antibodies. They have the same kind of binding loops. And of course, antibodies are blockbuster drugs, and other proteins are being developed as potential for treating various therapies or for diagnosing disease and so on. So these are a versatile target. They can bind a very versatile scaffold that can bind loads of targets, but aggregation is a real problem in developing these because they can aggregate at every stage of the process of developing a binder. So we start to collaborate. Oh, and look here, it's PEDS, a very highly cited review from our collaborator, Dasha Liposhek, on these kinds of scaffolds. So they were interested at Bristol Myers Squibb, Dasha's at Bristol Myers Squibb, in improving the solubility of the scaffold, actually, so you could have more flexibility in what you did with the loops. So what we found was that when we made different ADNectins that varied only, let's go back, that varied only in the loop sequence, this FG loop sequence, we got actually a really big change in inclusion body formation. So here you can see on the left is a loop. We have 3 residues changed that are shown in red. And here we have only 10% soluble protein, whereas with this loop sequence, we have 72% soluble protein. So that's interesting. Now this is in a loop, a solvent-exposed loop, and we found that this solubility was not correlated with global protein stability. The melting temperatures of all these variants are typically over 70 degrees. It's a very stable protein. So then we looked more at what the loops are doing. And so what Kyle did here is he modeled the stability with kinematic loop closure algorithm in Rosetta, and modeled many thousands, between 10,000 typically to 100,000 to test it further, conformations of the loop, and calculated the energy of the loop. And so here we have the average loop energy, and you can see that it correlates with, as the loop gets less stable, we get more inclusion body formation. We have some outliers here, and these actually come to what Bruno was talking about with edge strands. These are arginines in edge strands that may be acting as gatekeepers. If we take those into account, we get a better correlation. So if we're thinking about stability, stability is generally thought to affect aggregation. This is local stability rather than global stability. But if we think about what else might modulate this, because we have a lot of scatter here, of course, the aggregation propensity comes to mind because of these gatekeepers. So we then looked at what do aggregation predictors say. And so this is the Zaggregator score, which is an aggregation predictor from the Vendor-Scolo group. And you can see that as the aggregation score increases, so increased aggregation propensity, more of the adnectin is in an inclusion body. So here we have a fairly decent correlation as well. So we have local stability and aggregation propensity, and it turns out aggregation propensity and local stability are not correlated with each other. So when we combine these two features, we find that we can account for most of the solubility for adnectins. And so that's encouraging. It's a step in the right direction. I do think this kind of thing is can be highly protein dependent, but I also think what I want to point out here is that native stability and native aggregation is, I think, something we all have to think about if we're trying to design proteins. Okay, so how can we design increased solubility? So here we have, again, the structure of adnectin, and I'm highlighting here the F strand and the G strand in this beta sandwich structure, and here's the FG loop. So we wanted to improve the scaffold, and we looked at what the aggregation propensity was as a function of the primary sequence, and blue is soluble regions, red is aggregation-prone regions. And what you see here is that the F strand is a very highly aggregation-prone stretch. It's a central beta strand. They fairly often come up that way in folded proteins. So we thought this was a region to try to perhaps improve the stability, the solubility. So what we did was predict the solubility for a large data set of homologs of adnectins. So in the conserved domain database, there are over 740 of these. And you can group them according to which ones are predicted to have the lowest solubility beta strand, F, and which has the highest solubility strand, F. And what you see here is that we have hydrophobic residues at every other position prominently in both of these logos. This is the top 100, sorry, the lowest solubility 100, highest solubility 100. So we keep these hydrophobic core residues, which here are tyrosine, isoleucine, valine, pointing into the core. But the residues that are pointing out to the solvent that are in between these are markedly different. So we have more glues and arginines in these solvent-exposed positions. So we tried a number of these and got rather mixed results. Solubility did increase quite often when we put these residues in. This, by the way, is not an edge strand. But what we found is that V75R did a really good job at improving solubility. And when we then substituted a bunch of different loop sequences that we tested in the original scaffold that had a range of solubilities, you could see that the solubility increases in the V75R scaffold. So that was great. We got a more stable variant. And this was from inclusion body formation. We found that the folded protein was also, purified protein was also more soluble. Okay, so now, why are things more soluble? Can we understand the nature of aggregation? So for this, we turn to amide exchange. And in amide exchange, you measure when you dissolve a protein in D2O, how the backbone amides exchange with solvent, with the deuterium. And so the signals for the peaks disappear with time when you dissolve the protein in D2O. So if we look here at isoleucine 70 in the F strand, and here, this is in the G strand, it's hydrogen bonding partner. So I'm just going to go back for a second. So we're looking at these two. You'll note here that there's hydrogen bonding between isoleucine 70 and isoleucine 90. So you would think they should be protected together, right? This is a nice looking anti-parallel beta hairpin region. Well, not hairpin, but beta strand region, beta sheet region. The loop's too long to be a hairpin, but nevertheless, these guys should be hydrogen bonded. We find that isoleucine 70 doesn't exchange, so it's very protected in the wild type. But isoleucine 90, which is in the edge strand, in this G strand, is exchanging, is not as protected as it should be. So beta strand G is underprotected, and that was true also for other residues in the G strand. And remarkably, in V75R, which is more soluble, but not thermodynamically altered in stability, in its global stability, now the G strand is protected. So this is an interesting thing to think about. Oh, why would we have the F strand protected and the G strand unprotected in the more aggregation-prone wild type, and then we increase the protection in V75R? So our model for this is that somehow we're opening this part up to exchange without increasing exchange here. So this could be caused by self-association. So the protein sits there usually fairly cheerfully in solution. We can see this altered exchange in clear solutions that are highly concentrated. So this is some kind of transient association where you have a protected monomer, the protein self-associates, and then there's something that occurs in that self-associated state that allows this G strand to become exchangeable. So we're starting to get clues about what the self-association mechanism must be, because this is a particularly pronounced change. The rest of the protein has largely unaffected exchange, although we do think there are some other aggregation-prone regions there as well. But this was the most dramatic. Okay, now I'll come back to that because I actually think proteins, well, you can think about it. I'm not going to give you the punchline. You can think about this and tell me your opinion because I'm very curious. Okay, so we're starting to see where the association happens here, and this may also be relevant in cells, not just with the purified protein. This was done with purified protein. Okay, now SOD. So we've been interested for a long time in understanding how familial mutations in SOD that cause ALS, how those might cause disease. And if you look here, this is the dimer form of the protein, the mature holo form. It binds zinc and a copper. This is what makes it so stable. It also has a disulfide. Turns out there are many, many mutations throughout the protein sequence. The mutations are shown here in red that can cause disease. And for since 1993, people have been trying to understand this. We still don't understand it fully. There's a lot of evidence that mutations promote toxic misfolding and aggregation. The question is how exactly? And if you look at the mutations, what you see is that they tend to have different average disease durations that are quite variable. So A4V has an average duration of 1 year. It's a very rapidly progressive, terrible disease. In other cases, you can have a long duration. G37R also has a long duration. So we know there's this trend, and we'd like to understand how different forms of the protein might misfold and aggregate. Here I'm showing you the holoprotein. What I'm going to talk about today, we've done work on different forms of the protein, is I'm going to focus on the reduced apoprotein, which is actually just a monomer with no bound metals and the disulfide reduced. And that one is of interest because it's marginally stable. So this is a study we did a long time ago where we looked at reduced apo-SOD. So it's apo because it has no metals, and the disulfide bond is reduced, so 2SH, 2 thiols. And what we found was if we look at the stability, which is normalized here and organized from most stable variant to least stable variant in the reduced apo form, and the wild type is here. So not every mutation is destabilizing in this immature form. If you compare that with the disease duration, you get no correlation, right? The magenta bars are all over the place. So we don't have an answer from that, just stability. But we do see that as stability decreases, aggregation tends to increase. So that is the purple line here. And I say tends because there's a lot of variation here. So clearly other things are important for the aggregation as well. What are those? So we again turn to NMR to characterize the reduced apoprotein. So here it is, structure represented as a monomer. And here we have plotted the order parameter that you can get from chemical shifts versus primary sequence. And what you see here for the holoprotein is in red is that we have a high order parameter throughout, somewhat lower in the loop regions. And in the reduced apoprotein, we have considerably lower order in the loops, and particularly in this long loop here, loop 4, which is normally forms the disulfide interface, the dimer interface, and also has part of the disulfide bond. And this loop 7, which we call the electrostatic loop. So maybe I'll just go back there for a second. So just to orient you, here's this long dimer interface loop, and here's the electrostatic loop. They're going to come up again. So in terms of the reduced apomonomer, it's marginally stable globally, thermodynamically. I'm not showing you the data for that. And from further NMR analyses, we know the beta strands are intact, but these loops are flexible. So can we do more than just look at an order parameter? Here we use different kinds of experiments to detect rarely populated states. So this here is a chemical exchange saturation transfer experiment, where you can see that although you might have predominantly monomer in solution, you can see that there's transient formation of a native dimer. And the way you see that is in the CEST profile, you see a dip, where you have a dip at the chemical shift that's characteristic for the monomer, and another one at the chemical shift that's characteristic of the dimer. And this chemical shift is actually the same in the reduced apo as it is in the holoprotein. And we can look at nitrogen nuclei, carbonyl, and C alpha, and we can see what that dimer interface looks like. So it's very native-like. We also see by these methods evidence for transient native helix formation, and this is in that electrostatic loop, loop 7, we're going to come back to. Mostly, though, that loop is not well structured. And then we see, interestingly, so though we see these tendencies for the native protein to go to a more mature, for the native reduced monomer to go to a more mature state, we also see non-native associations. And these are mapped onto the structure here using CPMG experiments, and they show up as concentration dependent, and they have different temperature dependencies. And we see two kinds of dimer. We see a symmetric non-native dimer and an asymmetric non-native dimer. You can see here we've got a bunch of loops in the interface. This is shown better in this next slide. So here we have our native dimer. So here you see the N and the C termini are beside each other in the two subunits, so it looks like this. In this non-native symmetric dimer, what happens is you rotate around 180 degrees around this axis and flip this subunit over. So now the N and the C are down here. Here's the nitrogen and the carbon. So we do this. So it's a squishy interface. And you can also get an asymmetric dimer where now you rotate and you flip the whole subunit over. So now you've got different interfaces that give you more sticky spots to continue assembling. And one thing to note here is that both the dimer interface loop and the electrostatic loop that I highlighted before that are disordered are in that interface. So we also looked at mutations. I don't have time to go into that. And what we saw in general is that mutations tend to hinder going to these mature conformations, and they alter these processes as well. And they can also create opening of these edge strands, beta 5, beta 6. I'll come back to those. Okay, so this tells us what happens with purified protein. What would happen in inclusion bodies? So we looked at that next. And so we looked at a variety of mutants that are shown here in magenta. So we looked at ones in the dimer interface, A4V, that has that short disease duration, V148I and G, also in the dimer interface. So we made multiple mutations at different positions. And then we looked at some glycine mutations that change charge, so go to arginine or to aspartic acid. Here's another aspartic acid and an arginine, or to other mutations. So they're spread around on the structure. And just to orient you again here, we have the dimer interface loop, loop 4. Our electrostatic loop, which was blue before, is now red. And also loop 6 is going to be important. So what we found is that the mutations promote inclusion body formation. So this is, again, soluble in pellet. So the mutants are in the pellet, and they're in the reduced, apo-reduced form in the pellet. We found that inclusion body formation does correlate with stability. So as you get more stable, higher melting temperature, you have less inclusion body. And surprisingly, we saw that the proportion that was aggregating was also associated with the delta G of dissociation for the dimer. So this is now for with the disulfide intact, when it's formed, the apoprotein tends to dimerize. And so we measured its dissociation. And we found that as dissociation becomes more favorable and we have more monomer formed, we have more insoluble protein. So that's kind of interesting because, of course, we're looking at reduced apo, but we have other data that suggests that mutations effects propagate to the interface, and maybe the interface is just messed up. Okay, so now we used amide exchange to look at higher resolution at what's going on in the inclusion bodies. So this is quenched amide exchange, where we put the inclusion bodies into D2O. And so here in blue, we have all the amides that we can see when we dissolve the inclusion bodies. If we first exchange the proteins with D2O and then dissolve them, we can see what amides are protected. And you can see there's a lot of blue peaks here. There's a lot of protection. And this shows the residues that are assigned in blue. We know what almost all these residues are. And so we have a lot of probes of the inclusion body structure. So we were excited about this because there's not much known, as I said, about high resolution structures in inclusion bodies. And here we mapped, so we thought, okay, we should see differences with the mutants. So here I've plotted the mutants, and generally what you can see is that we have lots of amides protected. So a thing to note here is where we have gaps, we have no data. So some of those peaks are overlapped. So this is for well-resolved peaks. So we have probes throughout the structure and no information in these regions of gaps. So basically, almost all the amides are somewhat protected. Many are substantially protected. You can see here on the structure, they're all throughout the protein. So we have extensive exchange. And our charge mutants really didn't do very much. So the mutants tend to have very similar patterns. You can sometimes see some hints of local changes, but the charge didn't make dramatic changes. Okay, so let's back up a step. So I want to show you some data that was acquired some years ago in the Riek and Eisenberg groups. And so this was more like what we were expecting to see. So they looked at also marginally stable proteins, and here they have protonated spectra, and here they show the protected amides. So for this helical protein, it turned out that just this little stretch is protected. For this protein, it's only these few residues in a helix. And for this beta protein, it's 3 strands that are contiguous in the primary sequence. And in fact, if you're familiar with Eisenberg's work, he has a ZipperDB program that predicts amyloidogenicity. And so these regions were all predicted to form amyloid. So we thought, given that SOD was stable, that we'd see some marginally stable. And we'll come back to this. Eisenberg had suggested it would make amyloid based on peptide studies. We were expecting something like this. So as another piece of context, these amide exchange experiments have also been done on purified protein when it aggregates. And this is beta microglobulin, which is also associated with aggregation in disease. And it also has a beta sandwich fold. And here there's considerably more protection. So the central strands are protected, but A and G are not, the edge strands. Okay, so now, can we make sense of what we see? So we looked at this hard, and I'm not going to take you through all of this, but I'll highlight a few things. So here at the bottom is a study by Ivanova in the Eisenberg group, where they looked at peptides shown here in gray, which are in strands 2, 3, and loop 6, and the C terminus, and they found that they made amyloid. And so they make these steric zippers. And interestingly, this C terminus is predicted to aggregate a lot by a number of measures of aggregation, by hydrophobicity, by beta structure, and by various predictors, including Tango here. But these 3 or 4 regions don't account for all the protection that we see. So it turned out we needed to think about more than just unfolded proteins and little stretches aggregating. And we got some more insight when we started to compare what we saw with what we saw by NMR for the soluble protein. So here we have the native dimer, and you can see that we have protection in all the regions, including the loop 4, which is not protected by hydrogen bonds in reduced apo. That's the black bars here. And strand 8. So the natural dimer interface shows protection. So too does this, the symmetric non-native dimer, which also protects loop 6. So too does the asymmetric dimer, where we flipped it over, and now loop 7, which is the electrostatic loop, which is very polar, is also getting protection. And there's another crystallography study where they saw that native dimers could assemble into long filaments. And that happens by the electrostatic loop packing against these edge strands, beta 5 and beta 6. So that's what's here in this interface. And in addition, our native strands are protected. So what we conclude from this is that we're really seeing something very different from what the earlier study saw, in that we must be having multiple native-like structures aggregating. Okay, we actually think there are multiple aggregation-prone regions. So now it's interesting to also look at the mutant that didn't follow the general trend, which is V148G. So this mutation is in the final beta strand, and it turns out that its aggregation compared to the average, which is the dashed line, is changed. And in particular, the C terminus exchanges more readily, and loop 4 exchanges more readily. So here we have the C terminus and loop 4. Those two regions are basically in all those different dimers or in most of those dimers that we looked at. So we think that, yeah, and then at the same time, we see increased protection in loop 6 up here. So here we can see a change, and I think that's because V148G is interfering with a lot of processes. It's at a really key position, and in fact, it's right in that steric zipper region, and taking away the valine that packs against another peptide by mutating it to glycine would really interfere with that process. So we think this strong impact is visible because we've shifted the whole ensemble of aggregating species. And so the way we're thinking about aggregation, and I think this may be true for quite a lot of proteins, and there's evidence for it in P53, and I think also in ADNectins, which I'm not going to talk about, it's not ready yet, but we see extensive protection there too, is that you do have multiple aggregation-prone regions. And so here again, we have native dimer, which can assemble into these filaments that we're seeing in the ELAM structures. You can have non-native dimers, and maybe, you know, this zipper can go to, sorry, this C-terminal strand can kind of open up and make a zipper while the rest of the protein stays native-like. And so I think that by focusing on these details and by developing this amide exchange method, took a lot of work actually to get that method to really be robust, and it is now in our hands. I think we're starting to be able to understand this very complicated solubility and self-association landscape. And so as a summary, I think that solubility and self-association are increasingly understandable and therefore designable and definable using things like amide exchange. In terms of engineering proteins, there's a stability-solubility trade-off that was kind of unexpected. And I think native-like structure can be significant in both stable proteins and unstable proteins. You don't really necessarily get proteins that are naturally folded when they're unfolded tending to associate. Partially folded things, native-like things, are the things that tend to associate. And with that, I'd like to thank our wonderful team at Waterloo. They're just a delight to work with. We go through the hard times and the good times, but they're amazing in their teamwork. And I'm not really going to try to tease everybody apart here on the different projects. I'll just mention their names because there's actually a lot of discussion between the different projects. So here we have Aaron, Helen, Colleen, Ricky, John, Susan, Zach, Martha, and Avril, who are now alumni. This is the current group at Halloween. We won the contest. And of course, Jessica here too, and Christopher, who's in the group. And our collaborators, Freddie Colin's group we collaborated with on the screening for kinetic stability, Shachi with the coarse-grained modeling, which is ongoing, Dasha's group with the ADNectins, and on the SOD NMR with Lewis Kay's group. And of course, the funding agencies and Apprentice, which is training many of the people, and we have many of the people here in the audience too, I think. And thank you all for your attention. Okay, so I stop sharing, right?
All right, thank you, Liz, for a very beautiful presentation with lots of excellent data. Okay, so there's already a question for you.
Should I stop sharing? I should, yes, because then I will become larger on the screen. Not that I necessarily want to be larger on the screen, but there we go. Okay, let me just look at the question. Okay, is there a correlation between contact order and packing? Could you comment on packing interactions in threefold versus other less stable proteins? Thank you, Adam. Right, that's a good question. So threefoil, from what we can see in the structure, is really well packed. And when we tried to find stabilizing mutations, there were no holes. It was really well packed. And it seems that ricin superfamily, which is one of the superfamilies that makes this topology, this beta trefoil topology, those ricins, I think, fairly often are quite well packed. And a number of them are actually quite stable too. In these seed proteins, actually turns out there's ricins there that are very stable. By contrast, histactylin is certainly less well packed. So that's another beta trefoil. And actually, it has, I didn't talk about it, but it has a myristyl group that sits in the hydrophobic core. So it actually has fewer long-range contacts and more short-range contacts. And those short-range contacts are what bring its kinetic folding barrier down. Now, its folding barrier is less, but its thermodynamic stability is higher. So I think you cannot, it's another important thing to think about, you cannot assume kinetic stability and thermodynamic stability are going to be the same. They're different things. They often can be connected, but that's not a given. So the point is you can make a really stable protein with a rather poorly packed core. In the case of SOD, so it unfolds in the reduced apoform. It folds slowly and unfolds quickly because it's only marginally stable. And actually, its structure, it's a beta sandwich that kind of opens up. So I think that when you mess with a core, of course, you can destabilize a protein. I do think that correlations of stability with packing density are substantial. So, you know, that's part of various aggregation and stability, both aggregation but mostly stability predictors. They do look at how well packed the core is. And I think that getting something wrong in the core, like truncating a hydrophobic residue or putting a charge group in there, is really going to mess the core up. And we know a lot about that. So I hope that answers your question. So I think we can do a little better in the core. And I think particularly when we have hydrophobic residues, they're kind of squishy, and often they'll find a solution. But getting the fine details right is still hard. Okay. Suba. Hi, Suba. Are all the predictors used for predicting stability use structural information, or do they rely on sequence data alone? So yes, there's a wide variety of predictors, and they incorporate sequence data to varying extents. Some of them are only physical force field based, and some of them are more sequence based, and many of them are kind of hybrid, actually, or particularly the meta predictors include all these different kinds of algorithms. Yeah, and I think that there's still a lot to learn there, because being guaranteed of stability is not a thing. What do you think, Roberto?
I wouldn't have
discussion.
I wouldn't have said it better than you.
What's that?
You said it well.
Okay, okay, you agree. I think that's where the field really still is. Like, you know, you can make really stable proteins, and you know, the Baker Lab makes many of them, but they're doing a lot of things to the whole sequence. So then I think you can get there. So let me keep going down. Bruno, perhaps I missed it. Did you get a clear idea of the structure was in inclusion bodies and what were the biggest structural differences with the soluble protein? So no, you didn't miss it. So I was putting it out there because I wanted to see what you guys thought too. I would say, so the big surprise was we thought it would be unfolded protein with amyloid stretches protected, just a few amyloid stretches protected. I think that we need to do more work because this is, of course, we're seeing the whole ensemble. And so I think that in these inclusion bodies, we do have multiple conformations. And, you know, getting high resolution data on that is something that we want to do. So I think that inclusion bodies in general undergo structural transitions, and that's something we're following up on too. When they sit around with time, they may start native-like and then transition to amyloid. There's certainly data that suggests that. And so this mixture shifts. And I think that, you know, we're going to try to do things like design mutants to deliberately alter the structure. At this point, I think that we wouldn't get so much protection if we had an unfolded protein. So we have to have some native-like structure. Some parts of the structure that are protected are absolutely things that should not be protected based on hydrophobicity, based on hydrogen bonding. So actually those two loops, loop 4 and loop 7, are very flexible in the reduced apolipoprotein protein. They should not be protected. But we know from these crystal structures that, for example, that those loops can go into an interface. And we see evidence from that, not just in crystal structures, but in solution. So what I think is that those loops are actually not generally as protected as the beta strands. And so I think they are making those weak interactions. And we can look in more detail at what the extent of protection is under different conditions. So in terms of defining the biggest structural differences with the soluble protein, I actually think they're not that big. I would propose that it's pretty native-like. But at this point, we can't totally pin it down. These data don't show us that. But another way to try to get at that would be with looking at amide exchange by mass spec, which we may also try to do.
And Liz, maybe
I would like to. We have the instrumentation to do it. There's Joelle. Thank you, Joelle. Does the consensus approach lend more weight to long-range interactions by its very nature than other methods of prediction? That's an interesting question. Does consensus give more weight to long range? I'm just trying to remember. So Doug Barrick did some work on this that was interesting. He was trying to figure out what consensus is doing. So he really tried to take that apart with some of his anchor and repeats. And also, I think, with some of David Baker's proteins that were designed. Now, the anchor and repeats don't have long range. He didn't get a great answer, is kind of the take home here. I think that consensus can certainly capture long range, right? Consensus also captures functional residues. So it captures not only structure, but also function. Anything the protein really needs for the organism to keep it would be captured there, I think. And sometimes function and long range structural things will be at odds, and sometimes they'll be aligned. Often functional regions are unstable, but that's not always true. So it's a really complicated question. I do think consensus has the ability to capture long range, but Doug certainly also saw that it can capture short range. I think we need to learn more about that.
And Liz, can I ask you a question?
Please.
It's regarding the ADNectin V75R mutant. Do you think that that arginine side chain is somehow interacting with the FG loop in a way that stabilizes the outer strand and prevents it from, dissociating from the middle one? And that's how you protect the amide.
I think the arginine is interacting with the loop and therefore stabilizing that strand. I not necessarily. I think that the charge composition in that region is important. Certainly the loops sequences that we have vary in their charge. And if you so that arginine doesn't really help. You may have noticed there were some red and blue dots that you might have commented on before. Anyway, there are red and blue dots. The red and blue dots that didn't follow that correlation, the loop sequences that were shown in red and blue dots had net charge of that was either +1 for another arginine, an excess of arginine in the loop, or -1. So I do think that that arginine is kind of busting up a site that is prone to self-associate. And if you have another charged residue nearby, the arginine isn't as effective because that other charged thing is already busting it a bit. Unfortunately, we don't have probes in the loop. We really tried. We actually don't have assignments in the loop. We think it's self-associating. And in fact, in design, one of the big problems is that loops are often functional, designed to interact with something, and sites of association, right? So it's a big issue. So we're a little bit blind there. That said, so I think what's happening is, in those cases so far, it's too early to say. We need to look at more mutants, and Anna's doing that with Dahlia. And oh, I went all the way around and I didn't say the individual people. They're still here. No, I stopped sharing. Oh, of course. So the inclusion body work was Dahlia, who's in the CREATE program, and Mike, who's in the CREATE program. And they're working with Anna, who's not in the, not in the CREATE apprentice program. So they did a lot of work, and it's ongoing to try to analyze. We're looking at ADNectin mutants as well with the amide exchange. And I think it's too early to say, but it's a really great question. I would love to know the answer. Yeah. Okay. Don't know what that loop is doing. I wish we weren't so blind. It's nasty both in the purified form and in the inclusion body form when we bust the inclusion body.
Oh yeah, I bet. Okay, thank you, Liz. So I wanted to thank you again for your presentation. Please join me in thanking Dr. Meiering for her very nice and thorough presentation. And before I let you go, I just wanted to, again, thank everybody for their participation, for asking questions, for attending. And this will be the final webinar in this series. And I wish you all a good day and hope to meet you in the future. Thank you, everybody.